A Rational Approach to Antioxidant Therapy and Vitamin E. Written by Mark Hyman, Joseph Pizzorno, and Andrew Weil. Published in Alternative Therapies in Health and Medicine, 2005. The recent meta-analysis and media coverage surrounding the risks and benefits of vitamin E repeat a common failure of physicians and media. The sensationalization of medical research reports combined with lack of context and nuance that leaves practicing physicians, patients, and consumers confused. The need for health professionals to educate, inform, and provide guidance, not reflexively react to the shifting sands of scientific discovery or fail to understand the importance of considering new findings in the context of all levels of data and applying the rule of reasonableness is stressed. Interpreting studies, the importance of context and the role of the scientist. The recent meta-analysis and media coverage surrounding the risks and benefits of vitamin E repeat a common failure of physicians and media. The sensationalization of medical research reports combined with the lack of context and nuance that leaves practicing physicians, patients, and consumers confused. This is a repeat from the abstract. Fortunately, a more dispassionate and rational view, placing the current meta-analysis in the context of a broader literature on vitamin E and antioxidants, can be a better guide to recommendations and policy. Medical science is imprecise, and the tools of analysis and research are imperfect despite our best intentions. No statistical model or research methodology can accurately predict the outcome in all humans who vary greatly in habits and genes. Medicine has, in recent years, been humbled in its hunger to help by its mistakes, including hormone replacement therapy, which in the end hurt rather than helped the heart, the lipids-only hypothesis of cardiovascular disease that ignores the important role of inflammation, and the belated, sober end to our love affair with COX-2 inhibitors. Uncertainty is common in medicine. In the fact of uncertainty, we must take a broader view. I think they meant phase. Step back from the canvas of individual studies and meta-analyses and view the landscape of medicine and scientific inquiry as a whole, while understanding the limitations and value of different tools of research. We require all the tools in order to create sense and be sensible in a shifting sea of data points. The goal of seeking an informed balance overview is to help us create understanding from knowledge, to sort through the facts and place them in the context of biologic principles, against the backdrop of all we know. Then we ask ourselves if the new data fit into the landscape or appear like a polar bear in a desert. We must also recognize that the questions we ask, how we ask them, and why all inform the answers we receive. The responsibility of the scientist is to filter the research, place it in context, provide hope where appropriate, and caution where necessary, but most importantly, to be an informed, measured guide to a public seeking to gain health and ameliorate suffering. What can we learn from the recent example of vitamin E? There are a number of limitations from the recent vitamin E meta-analysis and opportunities to refocus our research strategy. Using long-term randomized trials and meta-analyses for the study of lifestyle interventions, diet, and individual nutrients is fraught with difficulty in design and effective separation of the plethora of their biologic effects. We may in fact be asking the wrong question. Asking if high-dose vitamin E is helpful or harmful may not provide the information we seek. We may better ask if restoring normal redox balance is protective, and if so, how can we best achieve that goal? Nutrients in general do not lend themselves to a research design that is best suited for pharmacological interventions because, unlike pharmaceuticals, nutrients are multifunctional substances built into our evolutionary design. There are many confounding factors in any randomized trial and meta-analysis. The vagaries in individual diet, habits, preclinical and clinical disease, genomic diversity of SNPs, SNPs, single nucleotide polymorphisms, the bioavailability and biological specificity of different forms of individual nutrients, and selection of the most effective combinations of nutrients make nutritional research through randomized trials and meta-analyses very problematic. Given that a methodologically sound, definitive, randomized, placebo-controlled trial has not been done and may never be done, the best we can currently do is take the best available evidence from basic science, studies of redox status, nutrient levels, trials with clearly measurable intermediate endpoints, relevant biomarkers, gene expression analysis, along with observational data in large-scale randomized trials, and then synthesize all the points of evidence into a biologically plausible theory, clinical guidelines, and when justified, public policy. This was not done. Meta-analysis confronts us with many methodological difficulties, including publication bias, heterogeneity of the trials, variations in quality, design, and purpose of the individual trials, as well as the fact that the findings are often not borne out in later randomized trials on the same subject. Laurier admonishes us that the popularity of meta-analysis may at least partly come from the fact that it makes life simpler and easier for reviewers as well as readers. However, oversimplification may lead to inappropriate conclusions. Furthermore, conclusions of many studies noted in abstracts of major journals are at odds with data published in the same paper. This oversimplification and conclusions inconsistent with the data were evident in the recent meta-analysis on vitamin E. Reviewing the review, errors in design and interpretation. With these caveats in mind, a brief examination of the basic science, observational data, randomized trials, and interventional studies with clear intermediate endpoints can provide a more rational context and framework for the recommendations about vitamin E, as well as a model for assessing the role of nutrients and other biological therapies in health and disease. The data in this meta-analysis pooled 135,967 subjects from 19 studies, which individually did not show any statistically significant increase in mortality. The 19 studies were selected from 2,170 studies. The authors concluded high dose, greater than 400 IUs per deciliter, I believe, per D, vitamin E supplements may increase all-cause mortality and should be avoided. However, the authors and the body of the paper provide adequate caution about overinterpretation of the results. They properly raise concerns related to publication bias, the small size of most of the included trials, the inclusion of patients with chronic disease limiting relevance to healthy adults, the concomitant use of other vitamins, including beta-carotene, that has shown an increase in mortality, they put morality, in smokers and drinkers, the lack of homogeneity of tocopherol isomers among studies, inconsistent use of synthetic all-rack alpha tocopherol or naturally occurring RRR alpha tocopherol, and the non-statistically significant reduction in mortality from doses less than 200 international units. The benefits for intermediate endpoints in many of the trials cited, they're citing Cambridge Heart Antioxidant Study, Chaos, SPACE, such as the reductions in non-fatal myocardial infarction by 70 to 77% were not discussed. These limitations were not included in media coverage nor taken into consideration by many of the expert commentators on the results. Other shortcomings of the meta-analysis by Miller et al. Are highlighted by a more careful review of the three flawed larger trials on which the conclusion that high-dose vitamin E supplements have a relative risk of death of 1.04 is based. These were the MRC/BHF11 heart protection study, Chaos 8, and AREDS 12, age-related eye disease study, report number 9, which represented 2/3 of the participants in the high-dose trials. The data of the other trials is less than convincing, and the interpretation and selective omission of key findings of the Chaos, AREDS, and MRC-BHF trials deserve closer scrutiny because they serve as the primary justification for the conclusions that vitamin E increases mortality. Of the 19 trials, 11 showed reduction in mortality, including 7 out of 8 low dose and 4 out of 11 high dose trials, which comprised only 30% of patients in the entire meta-analysis. It is also important to note that the all-cause mortality was available except for one study with combined high and low doses, that for the purposes of the meta-analysis, the doses were averaged. In addition, the statistical significance of the data is marginal. In the 11 high-dose trials, the risk difference was 39 per 10,000 persons, but the confidence interval was only 3 to 74 per 10,000 persons, and the risk ratio was 1.04, with the confidence interval of only 1.01 to 1.07, indicating that the risk may have been close to zero, even in the high-dose trials. The CHAOS investigators assessed the effect of RRR alpha tocopherol at 400 or 800 international units in 1,035 patients with ischemic heart disease. 967 more received a placebo for 510 days. The vitamin E supplementation reduced the risk of non-fatal myocardial infarction by 77%. There was a small, statistically non-significant increase in cardiovascular deaths in the treatment group. However, those patients were sicker at baseline. The deaths occurred very early in the study, and further analysis revealed that those patients were actually non-compliant with the vitamin E. Later mortality estimates from CHAOS came from a research letter, not a peer-reviewed study. 18 included data, 18 was a reference number, and included data after the study was officially ended, and thus subject to information bias due to a lack of complete reporting. The single largest trial, the MRC-BHF heart protection study, of 20,536 high-risk individuals using synthetic or racemic alpha tocopherol of 600 milligrams a day combined with 250 milligrams of vitamin C and 200 milligrams of beta-carotene, known to increase cancer risk in high-risk individuals. The authors concluded that the results effectively ruled out any substantial reductions or indeed increases in heart attacks, strokes, cancers, or other major adverse events during 5 years of use of these vitamins. Any benefit might have been mitigated by the use of potentially harmful synthetic beta-carotene and the synthetic racemic alpha tocopherol. While the study did measure serum levels of antioxidants, redox status was not assessed, leaving open the question of whether the antioxidants achieved their expected biologic effects. The AREDS trials are multiple, and the selection of the negative trials for inclusion in the analysis is perplexing, despite publication of conflicting results in the same issue of the Archives of Ophthalmology. Report number 9, included in the meta-analysis, showed no benefit in the reduction of cataracts from high-dose combined antioxidant therapy with beta-carotene, 15 milligrams, vitamin C, 500 milligrams, and vitamin E, 400 IU, either alone or combined with high-dose zinc, 80 milligrams, or copper, 2 milligrams, using an insoluble, non-absorbed form, cupric oxide. This is not unexpected because the primary mechanism of cataract formation appears to be glycation, not oxidation. Yet, an analysis of different endpoints from the same data in report number 8 showed a 34% reduction in age-related oxidative stress-mediated macular degeneration in 3,640 enrolled study participants aged 55 to 80 years, over 6.3 years. No statistically significant serious adverse effect was associated with any of the formulations in either analysis. Report number 11 estimated that if all persons at risk for AMD, some kind of macular degeneration, about 8 million, were to receive the antioxidants in the AREDS trial, the 300,000 of the 1.3 million at high risk for advanced AMD, I think it's age-related macular degeneration, would avoid that fate and provide considerable public health benefits. The studies cited either showed benefit in non-fatal intermediate endpoints, such as MI, myocardial infarction, macular degeneration, arteriosclerosis, angina, prostate and colon cancer, or at the very least, showed no harm. the conclusions in many studies do not accurately reflect the data. In the WAVE trial of hormone replacement and antioxidant interventions in postmenopausal women with coronary artery disease, the antioxidant treatment group was sicker, had more smokers and hypertensive, and less patients taking protective treatments with angiotensin-converting enzyme inhibitors or aspirin. So, it is not surprising that the vitamin treatment group showed a statistically non-significant trend toward worse cardiovascular outcomes. The authors found that neither the hormone nor antioxidant group showed benefit and suggested harm from both therapies, as noted in the conclusion. However, the lead author of the study, in a reply to a letter to the editor, conceded that these results do not prove that high-dose vitamin C and E supplements are harmful. The meta-analysis did not put its results in perspective. By reviewing the context of research on vitamin E, including the many positive observational and interventional studies, a large body of basic science research, and careful analysis of endpoints other than total mortality from the studies in the meta-analysis. It is important to note that many of the larger randomized trials showed statistically insignificant or barely significant benefit or harm. Also, randomized controlled trials on vitamin E and cardiovascular disease were subjected to meta-analysis in 3 other studies, and no significant increase in cardiac or all-cause mortality was found with doses of up to 800 IU per day. If vitamin E or the antioxidant theory is useful in preventing and treating disease, why do we not see a larger effect? It is that this question cannot be answered through randomized trials because of the enormous methodological challenge. Or is it that we must revisit the proposed value of antioxidants in secondary prevention? Any new study should be considered in the context of the best available evidence from animal studies, basic science, and observational and small and large interventional trials. The role of vitamin E in health and disease. The data on vitamin E collectively point to potentially important beneficial biologic effects that are at odds with the reported mortality data. While the basic science must also be cautiously used in making clinical assumptions, there are many reasons to hypothesize a useful role of vitamin E in promoting health as well as preventing and treating disease. A recent review on vitamin E outlined evidence that inhibits LDL oxidation, acts on coagulation, platelet aggregation, and endothelial aggregation, downregulates cellular adhesion molecules, inhibits release of interleukin-1 beta from lipopolysaccharide activated monocytes, inhibits protein kinase C, affecting a broad array of cell signaling molecules, and reduces inflammation and smooth muscle cell proliferation, induces apoptosis, and enhances cell-mediated immunity. Non-antioxidant activities of vitamin E include its influence on the activity of several other enzymes like PP2A, COX2, 5-lipoxygenase, nitric oxide synthase, NADPH oxidase, superoxide dimutase, phospholipase A2, and it also modulates the expression of genes that are involved in atherosclerosis, such as scavenger receptors, integrins, selectins, cytokines, cyclins. There are clinical trials indicating that vitamin E normalizes autonomic tone in diabetics, reduces risk for macular degeneration, prostate and colon cancer, reduces myocardial infarction, retards arteriosclerosis, and slows progression of neurodegenerative disease, as well as showing benefits in intermittent claudication, fibrocystic breast disease, premenstrual syndrome, osteoarthritis, and fertility. Because vitamin E is actually a mixture of 8 compounds, alpha, beta, gamma, and delta tocopherols, and alpha, beta, gamma, and delta tocotrienols. And because they are an integral part of cell signaling mechanisms in the integrated redox system, including ascorbate, carotenoids, R-lipoic acid, and glutathione, the ideal dosage, form, and blend of vitamin E and other antioxidants and cell signaling molecules is needed to clearly determine benefit or harm. Responsibility in science and the media. Unfortunately, answering these questions through randomized clinical trials may not be feasible because of the cost, time, and multiple variables required. We cannot assume that testing all nutrients as drugs in randomized clinical trials Even using a single agent with a single variable will be feasible, or, if done, will provide us with useful information with adequate external validity. Understanding the role of nutrients as part of the complex array of biologic response modifiers found in our diet and our environment should eventually bring more clarity and guidance for recommendations. Employing the linear reductionist model using varying study designs raises many questions regarding the validity of the data. Are the data in a population of patients with pre-existing cardiovascular, diabetes, and other chronic illnesses applicable to a healthy population interested in prevention? Are results based only on alpha tocopherols, racemic and RRR, synthetic and natural, relevant when diets contain mostly gamma tocopherols and the frequently used racemic form containing one alpha tocopherol may inactivate RRR alpha tocopherol? How do other forms of antioxidants and nutrients used in the studies affect the redox balance, signaling, and gene expression that determine harm or benefit? How do gene polymorphisms play a role in nutrient effects? Did the high-frequency polymorphism of endothelial nitric oxide synthase in the English patients in the CHAOS trial lead to the 77% reduction in non-fatal MI with vitamin E by improving endothelial function? Did differences in pre-treatment nutrient levels because of geographic location, dietary preferences, and nutritional status impact outcomes? Would patient stratification by clinical profile and assessment of biomarkers of vitamin E action, including effect on redox status, endothelial function, immune function, cell signaling, and gene expression, have provided insight on the possible risks and benefits with different population groups? New data is best interpreted in the light of existing recommendations and findings of independent and governmental bodies designed to guide policy. The recommendations from the Institute of Medicine, the U.S. Preventative Services Task Force, the Lewin Group, and literature reviews published in the Journal of the American Medical Association and the New England Journal of Medicine on vitamin therapy all contradict the findings of this meta-analysis. In fact, an independent analysis of the data on the clinical and economic impact of vitamins and health disease by the Lewin Group for Wyeth Consumer Healthcare found that the potential savings from multivitamin use in the Medicare population was $3.9 billion over 5 years from a reduction in cardiovascular and improved immune function and reduction in colon cancer with no attributable risk. In the light of this complex set of data, it is inappropriate for the media to publicize isolated findings of selected studies and propagate premature conclusions. It is even more concerning when the academic or practicing physician does not bring an appropriately open mind to consideration of complex biologic phenomena, namely oxidative stress in health and disease, and thereby avoid measured, thoughtful, and dispassionate discussion of any and all interventions related to this possible risk factor, whether natural or pharmacologic. Our duty is to educate, inform, and provide guidance, not reflexively react to the shifting sands of scientific discovery or fail to understand the importance of considering new findings in the context of all levels of data and applying the rule of reasonableness.